reproduction in organisms. Okay, so from this question you can expect, from this chapter you can expect 3 questions. And again, this chapter includes flower, pre-fertilization structure and events, pollination, double fertilization, post-fertilization events and structures. So let us start with the question. Microsporangium is generally surrounded by 4 wall layers. Okay, so which of the following 3 wall layers perform the function of protection and help in the dehiscence of anther to release the pollen? So options given are epidermis, tapetum, endothecium, epidermis, aerial, endothecium, epidermis, endodermis, mesocarp, epidermis, middle layer and endothecium. So here, let us see about the microsporangium. That is, microsporangium is having 4 wall layers as it is given. Okay, what are the layers? Let us see. Epidermis and endothecium, middle layers. So these 3 layers are involved in the function of protection. Clear? And the 4th layer, that is tapetum. This involved in nourishment, supplying nutrients. So here the question asked is protection. Protection is sponsored by epidermis, endothecium and middle layers. Clear? And the second question is, the above diagram refers to the TS of anther. Identify 1 to 5. So this diagram is given in your NCERT book and 5 parts are to be identified. Clear? So here I have given the exact picture which is given in your NCERT book, and this is connective. Okay, so this is connective, and again let us see, connective is given here, number 1. So number 1 here is sporogenous tissue. Sporogenous tissue. And here number 2 is tapetum. Just now I explained about the role of tapetum. Okay, which give nourishment. Okay, and the 3rd layer, 3, is again you see in this epidermis. That is the outermost layer. Clear? And number 4, this one is middle layer. Okay, and the remaining 5, last one is endothecium. So 4 layers and sporogenous tissue are given. Clear? So here in this one, sporogenous tissue, tapetum, epidermis, middle layer, endothecium. Option A is the right answer. Okay. So 3rd question, which of the following has the least pollen viability? So what do you mean by viability? How long it will be alive? How long it will have the potential of involving in fertilization? That is called as viability. So you know very well that the pollen grain, that is the characteristic feature of pollen, are it has 25 to 50 micrometer diameter. Clear? And it has 2 layers. It has 2 layers. Outer layer is called as exine. This exine layer is made up of sporopollenin. Sporopollenin. So often the question will be asked about this sporopollenin. Okay, and this is resistant to the specific feature of this sporopollenin material is this is having resistance against acid, alkali, and enzymes. No enzyme can digest this sporopollenin. Okay, and this pollen is having a germ pore, that is this exine. This exine is having a germ pore where this sporopollenin is absent. Question can be asked. Sporopollenin is absent in germ pore. That is a small pore which is present in the outer layer of the pollen grain. Clear? And the second layer, this is the first layer, that is outer layer. Second layer is called as intine. Clear? So this is the inner wall which is made up of cellulose and pectin. Okay, so these pollen grains have lifespan. So for example, that is if you take mature pollen, it is made up of vegetative cell. And again, one more is generative cell. Okay, so let us see the viability. If you take rice and wheat, cereals like rice and wheat, they have just 30 minutes. So after 30 minutes, it will lose its capability of fertilization, involved in fertilization. Okay, and others, if you take Rosaceae and Leguminosae, okay, Solanaceae. So these plants are having viability for months. Clear? So based on the species, the viability of pollen grains vary. So here the least pollen viability is for cereals like wheat and rice, that is only 30 minutes. Clear? So again, the next question, fourth question, yeah, that is given here. So this is exine. Okay, and the innermost layer is intine. Clear? And again, I already explained you that the mature pollen grain will have two cells. Okay, that is generative cell and vegetative cell. Clear? And that's all about the, that is again, viability. You have to remember viability. That is lifespan, you can say. Okay, so lifespan of pollen grain varies. Clear? So fourth question, identify 1, 2, 3, 4, and 5 structures shown in the figure female gametophyte. So why I have given so many diagrams in this structure, in this chapter, it consists of so many diagrammatic representations which help you to identify the mechanisms involving in different processes. Okay, so here 1 to 5 we have to identify. So this is embryo sac. Okay, generally which is having 7 cells with 8 nucleus. Okay, totally 7 cells are present with 8 nucleus. It means that, see here, okay, exactly this structure is given here. Okay, this is challicel N. Challicel N is having 3 antipodal cells. So number 1 is antipodal cells. How many in number? Antipodal cells. How many in number? 3. Okay, and 2, if you take 2, this is central cell. So the size of central cell is actually big, which is having 2 nucleus, 2 polar nuclei are present. Okay, so totally 3 antipodal cells. Number 2 is central cell, which is having number 3 polar nuclei. How many? 2 polar nuclei are present. Okay, and again, so what is this finger-like projection? Yeah, 3 we have completed now. 4, let us see what are 4 synergids. So what is the role of this synergids? So again, these are also 2 cells which help in fertilization process. So the pollen tube enters into the, that is releases the pollen grain, that is male gametophyte, into synergids. Okay, so that is the cytoplasm of synergids receive 2 male gametes. One fuses with the egg which is present here. Okay, this is micropylar end. This is micropylar end. And this is challicel end. So you have to remember that the egg is present in micropylar end, and the pollen tube enters through the micropylar end and enters and delivers the male gametophyte into the cytoplasm of synergid, and this male gametophyte again fuse with the egg. One male gametophyte fuse with the egg. Actually, the pollen tube delivers two male gametophytes. One fuses with the egg and other one male gamete fuses with the polar nuclei. So two fertilization occurs. This is called as double fertilization. Is it clear? So this is cell end. In cell end, 3 antipodal cells are present, and the central big cell is called as central cell, which is having 2 nuclei, which are called as polar nuclei. Clear? And again, in the micropylar end, we are having 2 synergid cells and 1 egg cell. So totally, how many cells are present? 3, 5, 6. Sorry. How many nucleus are present? Each cell is having 1 nucleus. So in antipodal cells, 3 nuclei will be there, and here polar nuclei 2, and egg cell will have 1 nuclei, and synergids will have 2 nuclei. Clear? So totally, how many nuclei are there? 8 nuclei are there, and number of cells, if you count, 3 at the cell end, 3 at the cell end, and 3 at the micropylar end. Clear? And central cell 1. So totally 7 celled with 8 nucleus. So this is the characteristic feature of embryo sac. Clear? So now I think it is clear. That is, number 1 is antipodal cell, number 2 central cell, number 3 polar nuclei, number 4 synergids, number 5 filiform apparatus. So filiform apparatus through which that is helping in the entry of pollen tube. Okay, number 5 is filiform apparatus. So which one is correct here? 4 synergids, number 5 antipodal, central, polar nuclei, synergids, filiform apparatus. B is the right answer. Is it clear? Okay. So pollen grains can be stored in liquid nitrogen at minus 196 degrees C. So why the storage of pollen grain is important? Because some species having threat of extinction. So we can store the seeds, pollen grains and seeds actually, okay, with the help of this liquid nitrogen, and we can maintain even semen, we can maintain with the help of this liquid nitrogen, and we can maintain minus 196 degrees C. Why? How we can store means by inhibiting the metabolic activity. Okay. 6th question, which of the following points is incorrect about sporopollenin? 6th one, which of the following points are incorrect about the sporopollenin? Okay, it is one of the most resistant organic material known. Yes, we have to find out the incorrect statement. So already I told you that this is organic material which is having resistance against acid, temperature, enzymes and alkali. So it can withstand high temperature and strong acids and alkali. Yes, this is also true. 2 enzymes that degrade sporopollenin are known so far. No, this is wrong. No enzyme can degrade sporopollenin. Pollen grains are well preserved as fossils because of the presence of sporopollenin. So here the wrong answer, the wrong statement given, incorrect statement given about sporopollenin is enzymes cannot degrade sporopollenin. 7th question, in animals exhibiting internal fertilization. In animals exhibiting internal fertilization, there is high synchrony between 2 sexes, no need of mating. Mostly the male gamete is motile, vulnerable to predators. So in animals exhibiting internal fertilization means the fertilization occurs inside the organism. For example, human being, inside the organism. So if you take for internal fertilization, we can take human as an example. So here what is happening? The egg is staying at fallopian tube. Clear? And the male gamete, sperm, sperm is motile. It reaches fallopian tube by traveling. Okay, so here Which one is correct then? Mostly the male gamete is motile. Clear? So here there is high synchrony between two sexes are not needed. And no need of mating, that is population is needed for fertilization. Okay, this is not at all related to internal fertilization. That is vulnerable to predators, means predators will destroy the gametes. That is not possible in internal fertilization, as the male gamete or that is male gametes are delivered inside the body of the female. So there is no chance of predators attack. Clear? And the next question is, gatenogamy is functionally cross-pollination involving a pollinating agent. Functionally, cross-pollination involving a pollinating agent. Genetically, it is similar to autogamy since the pollen grains come from the same plant. Functionally and genetically, autogamy. Both A and B are correct. So here, let us see about the types of pollination. So this is one type of pollination. Okay, so pollination types. Clear? So the first one is autogamy. So what do you mean by autogamy? Here, the pollination occurs between flower, that is the anther and meeting of egg and anther happens in the same flower. That is shedding of anther to the stigma. You can have like that, anther to the stigma of same flower. Clear? So that is called as pollination, and autogamy means the same flower will have the stigma and the anther of the same flower will be reaching the stigma of the same flower. That is called as autogamy. And again, here that is at the same time it should happen. Synchronization is must. That is pollen release and stigma receptivity, we can say. Okay, both should synchronize, occur at same time. Occur at same time, so that pollination is possible. Clear? And again, in this there are two types, that is chasmogamy. Clear? Chasmogamous. What do you mean by chasmogamous? For example, it happens in viola and oxalis. So here the flower is open. Okay, that is the that we can see anther and stigma here. But the second type in autogamy is cleistogamous. So here the bud won't open. The bud is closed. Okay, so as there is no need of pollinators then, no need of pollinators. But in the first class, they need pollinators. Need pollinators. Question can be asked. That is, chasmogamous flowers need pollinators, but here cleistogamous flowers, they are having closed buds. They won't open. So that inside the bud, what is happening? Pollination is happening. So there is no need of pollinators. So inside the bud, what are they? What are there? That is anther and stigma. Both are there. So there is no need of pollinators. The anther will meet the stigma. This is called as autogamy. So the second one is gatenogamy. The second type is, which is given in the gatenogamy. So here, what is the difference between gatenogamy and autogamy? This is otherwise called as cross-pollination. Cross-pollination. So here, autogamy, one flower, single flower is involving in the pollination process. In gatenogamy, in the same plant, but two different flowers. Two different flowers. So the anther of one flower is reaching the stigma of the another flower. That is called as gatenogamy. And again, here also pollinators are needed. Okay. So again, the third type is xenogamy. xenogamy. Here, the pollination happens between two different plants. So here, compulsorily, the plants are in need of pollinators. Is it clear now? Autogamy happening in the same flower. In that, two different types are there. One is opened flower and the other one is closed flower. Okay. And again, here, the second type is geitonogamy, that is cross-pollination. The same plant, two different flowers will involve in pollination process, which is called as cross-pollination. And again, xenogamy is also example for cross-pollination, which is occurring between two different plants. So here the question is, functionally, cross-pollination involving a pollinating agent. Definitely, cross-pollination requires pollinating agent. Genetically, it is similar to autogamy, since the pollen grains comes from the same plant. Yes, geitonogamy, it is cross-pollination, but genetically, the flowers will be same because the pollination occurring between the flowers which are present in the same plant. So genetically, they will be similar. Okay, functionally and genetically, autogamy. No, we can't say functionally and genetically autogamy. So both A and B are correct. Okay, so next question is, scion is grafted to a stalk. The quality of fruits can be determined. The quality of fruits can be determined by the genotype of. So this question is from reproduction in organism. So grafting, what do you mean by grafting? Combining of two different plants is called as grafting. Okay, so in grafting, two parts we are in need of. One is scion and the other one is stalk. So what do you mean by scion? Scion is the top part of the plant. Top part of the plant, which is having flowers. Which have flowers, you can have like that. Okay, so then what is stalk? Stalk is the bottom portion which is having root system. Bottom which has with roots, you can say. So here the question is, the quality of fruits can be determined by the genotype of. So the fruit quality depends on flowers. Clear? So here we are having the scion. That is, the flowers are present in the scion. So definitely the genotype of scion is responsible for the quality of fruits. So scion is responsible for the quality of fruits. Clear? So the correct option is scion. Yes, the ploidy levels of the cells of nucellus, megaspore mother cell, functional megaspore, and female gametophyte. Okay, that is the ploidy levels of the cells of nucellus, megaspore mother cell, the functional megaspore, and female gametophyte. So if you see up to this, the mitosis is happening. After this, meiosis is happening. You have to remember that. So it will be easy that you have to remember that due to mitosis, there is no reduction happens in the chromosomes. Okay, so here in these two, the number of chromosomes will be 2n, diploid. And the functional megaspore and the female gametophyte, they are haploid in nature. So these two are functional megaspore and female gametophyte. Both are same. Actually, they are haploid in nature, n. So here the order will be 2n, 2n, n, n is the right answer. Clear? Yeah. The largest cell in the embryo sac is, see, already I explained, totally 7 cells are there. Chalocell end has the 3 antipodal cells which are same in size, smaller in size. And again, in micropilar end, we are having 3 cells, 2 synergids and 1 egg cell. Clear? They are also small when compared to the central cell. See here the size of the central cell, which is having 2 polar nuclei. One cell having 2 nuclei. Clear? So the biggest cell in the embryo sac, this is the structure of embryo sac. So that in the structure, it is clear that the central cell is the biggest cell in embryo sac. Which type of association is found in between entomophilous flower and pollinating agent? Entomophilous flower. What do you mean by entomophilous flower? Entomophily, that is which requires insects for pollination. Okay, entomophilous flower, it got its name due to the need of insects for pollination process. So here the pollinating agents are insects. Okay, so what association is there in between the flower and this insects which are helping in pollination? Okay, that is mutualism. So what do you mean by mutualism? You know that both will get benefits. So both are getting benefited. So here if you take, here if you take flower, insect is helping in pollination. So what is the advantage? Flower getting from insect, pollination process is happening. Okay, and insects, they are getting nectar, nutrients from flower. Okay, so these two are having mutualism. Yes, the 13th question, again, this question is from Reproduction in Organism. Lifespan of parrot is. Lifespan of parrot is. So while talking about lifespan, you have to remember that the lifespan and size is not having any correlation. Okay, so if you take the size of parrot, it is very small, which is having 140 years of lifespan. But elephant, so let me give you elephant, which is having 60 to 90 years of lifespan, clear. And the second, you can take rose plant. So rose plant is having 5 to 7 years. And third one, if you take dog 20 to 30 years. And if you take butterfly only 7 to 14 days or 1 to 2 weeks, you can say. And if you take fruit fly, so fruit fly 1 month, 30 days. And rice plant, if you take rice plant 3 to 4 months, 3 to 4 months. Okay, and next one, banyan tree, that is up to 200 years. And if you take crow, that is having 15 years of lifespan, and cow 20 to 25 years. So question can be asked from this in different ways. Okay, 25 years. Banana tree 25 years again. Crocodile, if you take 60 years, and horse again 60 years of lifespan, and tortoise 100 to 150 years. Clear. So here the lifespan of parrot is 140 years. The term pollination signifies dehiscence of anther. What do you mean by dehiscence? Release of anther. Clear. The transfer of pollen grains from anther to stigma. So this is the correct definition for pollination. The transfer of pollen grains from anther to stigma. Okay. So next one is the transfer of pollen grains from anther to the stigma of the same flower. It is not needed. It is not. This is also pollination, but it is not necessarily needed. Formation of pollinia. Okay. So the next question is the study of formation, growth, and development of new individual from an egg is called. The study of formation, growth, and development of new individual from egg is called embryology. Okay. So egg meet with male gamete. Okay. First form zygote. Zygote form embryo. and from embryo, full organism will be developed. So there are so many events happening in this process. So each and every event involves so many factors. So study about these reactions, these processes comes under embryology. Okay. So find out the correct statement. Lifespan and size of the organism are correlated. No. Okay. Just now we saw that the elephant can live from 60 to 90 years, which is bigger in size, but the smaller parrot can live for 140 years. Size and lifespan of crow and parrot are same. No. Size is also vary. Crow is bigger than parrot, and the lifespan, if you take crow, 15 to 20 to 25 years. Yeah, sorry, 15 years for crow and parrot, 140 years. So nearly if you take almost 10 times greater for parrot, right? Banyan tree has much shorter lifespan. No, banyan tree is having 200 years, but mango tree, not that much. Reproduction is essential for continuity of life. Yes, this statement is correct. Reproduction is essential for the continuity of life. Which of the following statements is correct? Majority of plants use biotic agents for pollination. Yes. So what are the biotic agents involved? Air, water, that is wind. You can say wind, water. Sorry, abiotic. These are abiotic. So what are biotic agents then? Living organisms. So living organisms are biotic agents, and abiotic agents are non-living pollinating agents. So pollination by wind is more common among abiotic pollinations. Yes, definitely. Pollination by water is quite rare in flowering plants. Yes, only in aquatic plants this pollination agent will be water. Okay, so all are correct statements. Okay, repeated self-pollination over the generation produces. So self-pollination means, what do you mean by self-pollination? That is, the anther is transported to the stigma of same flower. So here, what will happen? There won't be any genetic combinations are possible. So automatically the same quality will be produced, same quality, or else the quality lesser than the parental will be obtained after many generations. So this is called as inbreeding depression. Okay, same quality or decreased quality, which is called as inbreeding depression. Yes. Yeah. So double fertilization involves fertilization of the egg by two male gametes. No, one egg will fuse with only one male gamete. Clear? Not with two male gametes. Fertilization of two eggs in the same embryo. Never the same embryo will have two eggs. It will have only one egg. Embryo sac by two sperms brought by one pollen tube. No. Fertilization of the egg and central cell by two sperms brought by different pollen tubes. No. Fertilization of the young egg and the central cell, two sperms with different pollen tubes. No. Fertilization of the egg and central cell, two sperms brought by same pollen tube. So let me explain the process here. So you very well know that is here, this is the primary endosperm nucleus, how it is formed. So how it is formed, what is called as double fertilization? So double fertilization means the fertilization occurs twice. Fertilization occurs twice. So here, how the fertilization occurring? That is generally the pollen tube enters synergy. And filiform apparatus guide this pollen tube to enter synergid cells. And after entering the synergid cells, it releases two male gametes. two male gametes are released, okay, by one pollen tube. And in these two male gametes, one fuse with egg. Okay, you can consider this as first fertilization. First fertilization. Clear? And again, we are having remaining one male gamete. Okay, so the second male gamete fuse with the central cell. Fuse with the central cell. So this male gamete is having one nucleus, which is haploid in nature, that is N. Okay, number of chromosomes N. And here in central cell, we are having two polar nuclei. Each are haploid in nature. Clear? So totally how many nuclei are fused? 3 nuclei. So this is called as triple fusion. Because this N plus N of polar nuclei. That forms 3N status, triploid. So primary endosperm nucleus is triploid in nature. So this forms, that is the second fertilization. Clear? So the second fertilization is responsible for endosperm. Clear? So the first fertilization leads to the formation of ovules. Clear? So this is the explanation for double fertilization. From this, so many questions can be framed. Okay, so in first fertilization, one male gamete is fused with one egg cell. Clear? And in the second fertilization, second male gamete, which is entering through the pollen tube, fused with the central cell. In central cell, two polar nuclei are there. So totally 3N triploid cell will be formed. That name is primary endosperm nucleus. Primary endosperm nucleus, PEN. So this is responsible for the formation of endosperm. Okay, clear. So total number of nuclei involved in double fertilization is involved in double fertilization is, just now we saw two male gametes. Clear? So from this concept, double fertilization, different questions can be framed. Two male gametes and one egg cell and two polar nuclei. So here the question given is total number of nuclei involved. So totally how many? 5 nuclei. If the question is asked about cells, how many cells? How many cells involved? 2 gametes, male gametes and one egg. 2 male, 1 egg and 1 central cell. Clear? So now the how many nuclei are involved? 5. Entry of pollen tube through micropyle is called as porogamy. Because it enters through the micropyle, which is a small pore, pore-like structure. So this process is called as porogamy. So which among the following is not a feature of pistil? So which among the following is not a feature of pistil? Monocarpellary, syncorpus, apocorpus, mitocorpus. So what do you mean by monocarpellary? So what do you mean by carpel actually, which is having ovary, style and stigma. So this is stigma, this is style and this is ovary. So altogether this is known as carpel. So in many flowers have this monocarpellary, that is one carpel. Okay, and multicarpellary plants are also there. Multicarpellary flowers, again, I have to say. Multicarpellary. What do you mean by multicarpellary? They are having more carpels. More carpels. So in some flowers, if you see, the carpels are, so this is one ovary. Multi ovaries means many ovaries will be there. Clear? So let me show two ovaries along here. Okay. So here they are separated. And stigma is here. Style is here. Stigma is here. This is called as apocarpus. Okay. But in some plants, the ovary, again, that is, if you take the structure, it will be different. Let me show you. See here, style and stigma, they are fused, right? So this is called as syncorpus. Okay, syncorpus. So these three, this is monocarpellary. Monocarpellary. So in dicarpellary, there are two types. One is apocarpus, second one is syncorpus. Either they are separately present or they are fused, like in syncorpus. Okay, so these three are the terms involved in the pistil. Okay. So in double fertilization, total number of male nuclei and total number of female nuclei involved are. So male nuclei 2, female nuclei 3. What are they? 1 egg and 2 polar nuclei. Clear? So here 2 and 3 respectively. Okay. So identify A, B, C in the given diagram. This is again from reproduction in organism. These are gametes, types of gametes, you can say, types of gametes. So gametes, that is both female and male may be same. If they are same, they are called as isogametes. See in A, these are isogametes of Cladophora and algae. Okay, so one is female gamete, one is male gamete. One is female gamete, one is male gamete. Both are similar in structure. So this type of gametes are called as isogametes, and the example is Cladophora. Okay, and if you see in the second diagram, this is male, female gamete, and this is male gamete. See the size, everything is different. So these are called as heterogametes. Example, fucus. Example, fucus, and this is heterogametes. Okay, and you know very well the structure of sperm and egg. So this is egg. So entirely different, right? This is having oval shape and this is having spiral shape. Okay, this is sperm, and exactly this is the gametes of human, female and male gametes of human being. Okay, so let us see the options given. So option A is isogametes. Isogametes, example, Cladophora. Yeah, isogametes, Cladophora. And the second one is heterogametes. C is human being. So this is wrong. A, isogametes, it should be. None is also wrong. A is the right answer. So the figure A is isogametes, B is heterogametes, and C is the gametes of human beings. And the last question, which of the following is false about emasculation? So why emasculation is needed? For example, even in genetics, you might have learnt about this, that is for artificial, yes, artificial fertilization. Fertilization or hybridization, you can say. So we have to separate the anther from the flower, male flower. Okay, so the removal of anther from male flower for using artificial fertilization is the process called emasculation. So during emasculation process, stigma is removed. No need to remove stigma. Okay, we should not actually, during emasculation, we have to take care that we should not disturb the female reproductive system. Clear? So emasculated flowers are bagged in order to prevent self-pollination. Yes. Emasculation is the removal of stamens before anther dehisces in bisexual. No need to remove. If you tie a bag, we can collect the anthers. Okay. So it is, yeah, it is one of the steps for artificial hybridization or fertilization. Yes, this is also right. So which one is wrong here? During emasculation, there is no correlation between stigma and emasculation. So this is the false statement given here. Okay. So that's